So the first assay we developed here at PHE was to use wild-type circulating virus. We used the Victoria 2020 strain for this. So that's actually, although the virus came out of Wuhan, the very first isolates that were available for use were actually coming out of Australia, just because of the way international agreements work. But to all intents and purposes, it's the same virus. There's one small change in spike between this and the Wuhan. So the PRNT, or plaque reduction neutralization test, was the first assay we developed, and then that became a refined, much more high throughput method as a focus reduction neutralization test. And I'll probably talk about microneutralization as I go forward, because FRNT is a bit of a mouthful. And if you're interested in these methods, you can find them in a Nature Protocols paper that was published earlier this year. And as well as developing that for looking at the prototype strain of COVID, we've also been using that assay for several of the variants now. And we're looking at VOIs or VUIs, depending where in the world you are, and also the variants of concern, the VOCs. So for those of you who aren't familiar, to do a neutralization test, you dilute antibody samples in media. You then incubate that with a constant amount of virus. You infect that into a susceptible cell line. After an appropriate amount of incubation, you fix those cells, and then you stain to either visualize plaques caused by the virus, and they're the holes on a blue background that you can see in one of those images there, or you can immunostain these tiny foci of infection in a microplate well, or if you're doing pseudo-neutralization, you might have a colorimetric or a luminescent or fluorescent label inside that pseudovirus that you can use to read off instead. So then we read whatever the output happens to be, whether that's foci or color change. We fit curves to the neutralization results, and we report those onwards. So the PRNT is really the gold standard for measuring neutralization assays. It's not a great high throughput method, but it's something we have a lot of experience with here at Porton on high containment viruses. So we can do all this at containment level 3 and 4. And that plate on the right, basically what you're looking at, if it's purple, it's a cell. And if it's a hole or white, that's where the virus has made a small hole in the monolayer. And by looking at the reduction, so on the bottom row, you can see a sera that is reducing that count down. So on the right-hand side, there's maybe a dozen spots where there would be 100 in the unneutralized controls. We can plot a curve through those reductions, and we can figure out what the median neutralizing dose of a given sera is. And this is a good measure of neutralizing antibody activity. And as well as looking at serum from people, you can also test therapeutic monoclonal antibodies, animal sera from preclinical studies. It's a very versatile assay. And unlike, say, ELISA, you don't need a conjugate, so it doesn't matter what species of sera you're using. You're purely interested in whether it can knock down the number of holes or plaques that you're making. So these are functional antibodies that are working physically on the virus. So the PRNT, it's a great assay, but it takes 7 days with COVID from setting up the cells in your plates to actually getting an answer. And each one of those plates gets you the answers for 2 sera. And if it goes out of range, you actually have to do it again. So it can be 2 weeks before you actually know your titer, which isn't really good enough. And you always lose a plate to having a reference and virus-only controls and those sorts of things. And to count those spots, although we haven't really looked and invested in computerized counting systems, it's still mainly done by people hunched over a light box, counting those little spots and typing them into Excel. So we refine that by performing the Microplaque, which is effectively stained foci. We do these in 96 well plates, which means we can straight away on a single plate run 6 samples plus a reference and the virus-only control. We can also have more counts per well in the unneutralized controls, which gives us a more high-resolution curve than a PRNT would. We're counting these spots using an Ellis spot counter, so the counting is now completely automated, and the count files are analyzed in Softmax Pro which fits a modified 4PL through the neutralization counts. And that whole assay now is 4 days. The actual foci are read 20 to 24 hours after setup. So the full 4 days is from seeding the cells to actually having data ready to report.